40 years of clinical trials focused on amyloid beta clearance have failed to produce disease-modifying results. These outcomes have led to a critical reassessment of the physical identity of the Alzheimer's plaque and whether the model of passive protein accumulation captures the full scope of the tissue remodeling occurring in the brain. When we look beneath the protein deposit, we find a highly organized fibrillar lattice. Watch closely as microglial enzymes are actively severing and densifying the brain's existing architecture into the structures shown here. This process is known as extracellular matrix remodeling. This fibrous network anchors and sustains neurons, and the activity of microglial enzymes within it provides a new focal point for understanding disease progression. If the primary pathology is a microglial-driven rearrangement of the matrix, clearing the secondary protein deposits would not be expected to halt the destruction of the underlying neural scaffold. The focus on deposits over matrix architecture dates back to a divergence in 1907. While Alois Alzheimer's neurofibrillary framework was widely adopted, a competing framework proposed by Oskar Fischer in Prague was largely set aside by the scientific community. Fischer based his findings on a 58-brain cohort, utilizing the Bielszowski-Silver stain to visualize the fibrillary proteins of the extracellular compartment. He named the resulting lesion spherotrichia cerebri multiplex, multiple spherical thread formations, defining the pathology as a structural matrix abnormality. In the 2020s, a new body of research led by investigators like Kropser and De Vries identified the microglial-mediated degradation of specialized extracellular structures known as perineuronal nets. This substrate forms what is now categorized as the seventh convergence axis. Modern transcriptomic data documenting the loss of perineuronal nets corresponds to the histological observations Fischer recorded in 1910. Both frameworks describe the same physical degradation of the cortical ground substance. The 2026 focus on perineuronal net integrity restores the foundational observations Fischer recorded in 1910, providing a molecular basis for his original histological framework. Fischer identified 4 specific points where his observations align with modern matrix biology. First is the nature of the plaque halo. Fischer wrote that the halo has the same structure as the surrounding cortical ground substance. This contrasts with the later interpretation of the halo as a diffuse accumulation of a foreign toxic protein species. Fischer observed that the surrounding tissues were arranged somewhat more closely. Multiplex immunofluorescence now shows microglial enzymes digesting structural proteins like aggrecan and tenascin R, causing the remaining fragments to condense locally. The halo represents a structural rearrangement of the existing perineuronal matrix, rather than the arrival of a foreign deposit. Fischer's second observation was a temporal sequence. In 1910, he outlined an 8-stage progression for the lesion, tracking it from an initial star formation through expanding spoke and wheel phases, ending as a mature fibrous ball. This chart tracks the biological mechanisms. As morphological stages evolve, matrix metalloproteinase activity rises. MMMP2, MMMP9, and ADAMTS4 actively sever the matrix. Fischer noted this remodeling occurred without classical reactive inflammation. Modern data confirms this. Enzyme expression spikes while leukocytic inflammatory markers remain flat. This is the cold microglial phenotype. Microglia execute a targeted enzymatic degradation program at the perineuronal net without recruiting peripheral white blood cells or causing gross tissue swelling. Because microglial matrix remodeling is an inherently non-inflammatory program, it would not be addressed by therapeutic strategies aimed at a classical inflammatory cascade. The third convergence is anatomical. In his study of 58 brains, Fischer mapped the highest concentration of lesions to the frontal lobe and the upper cortical layers, specifically layers 2, 3, and 4. He documented negative findings, noting an absolute absence of the lesion in white matter, the thalamus, and the cerebellum. Overlaying the 2024 mapping of parvalbumin-positive interneurons and their perineuronal nets reveals a perfect distribution match with Fischer's data in every region. This anatomical alignment indicates that both the historical and modern observations are focused on the same biological substrate. This regional precision maps the disease to a targeted structural failure of the parvalbumin interneuron matrix, matching the distribution Fischer observed in his initial cohort. The fourth convergence appears at the microscopic periphery of the lesion. Fischer recorded radially arranged club-shaped structures pointing outward from the lesion's edge. He identified these as swollen neuronal processes that resembled the severed ends of regenerating nerves. Modern neuroanatomy identifies these as the axosomatic terminations of parvalbumin interneurons. Watch as the protective perineuronal net is enzymatically digested. Without that scaffold, the interneuron retracts its processes, resulting in the swollen club-like ends Fischer described. This retraction is the mechanical consequence of losing the trophic and structural support provided by the intact extracellular matrix. The neuritic dystrophy identified throughout the 20th century is the physical collapse of parvalbumin interneurons following the destruction of their surrounding matrix. This cellular failure scales up to the clinical phenotype. Fischer linked these matrix lesions to a specific syndrome, presbyophrenic dementia. Presbyophrenia is characterized by rapid memory loss, aggressive confabulation, and hallucinations. Parvalbumin interneurons regulate gamma oscillations in the entorhinal cortex. As the perineuronal net degrades, these neurons lose their capacity to sustain the firing rates required for gamma waves. The oscillations desynchronize, leading to the electrical misfiring that manifests as confabulation and hallucination. At the perineuronal net, the microglial attempt to shield the interneuron via matrix digestion results in the immediate collapse of the circuit's electrical integrity. The clinical manifestation of the disease is a readout of parvalbumin interneuron electrical failure, driven by the collapse of the extracellular matrix. These convergences shift the focus of drug development. If the primary crisis is matrix destruction, modifying the disease requires looking beyond the clearance of secondary amyloid deposits. Disease-modifying strategies must target the preservation of the aggrecan lattice, the reinforcement of tenacin R anchoring canes, and the suppression of the enzymes responsible for severing the net. These interventions are aimed at halting the upstream progression of Fischer's 8-stage sequence. After a century of displacement, the field is returning to the framework established by its marginalized founder. Alzheimer's is a disease of the cortical matrix. The preservation of the parvalbumin interneuron perineural net is the substrate on which future therapeutic development must build.